Good evening, everyone, and welcome to this set of lectures on incredible topics which we cover in basic science and translational medicine from Stem Cell Center. I would like to introduce our speaker for today. I would like to thank Professor Rohit Kulkarni from coming all the way from Boston, the Joslin Institute, to give us this talk. Professor Rohit Kulkarni is indeed a very esteemed personality in the field of science, and he holds the Diabetes Research and Wellness Foundation Chair, Section Head of the Islet Cell and Regenerative Biology, and Associate Director of the Diabetes Research Center at Joslin. He's Professor of Medicine and at the Harvard Medical School and Associate Member of the Broad Institute at MIT, Harvard, and Principal Faculty at the Harvard Stem Cell Institute. He is indeed an endocrinologist by training and has now delved into basic science in almost all aspects of islet cell biology. He is recognized for his excellence in diabetes research by the Ernst Oppenheimer Award by the Endocrine Society, the Albert Renaud Prize by the European Association of Study of Diabetes, the ASD, and the Paul Lacey Medal and Award, which, of course, as you know, Paul Lacey is the pioneer for islet cell transplantation. He is an elected member of the American Association of Clinical Investigation, Association of American Physicians, and American Association for Advancement of Science. His lab is interested in investigating mRNA modifications, stem cell biology, and organ crosstalk in the long-term goal of finding cures for diabetes and obesity. Thank you, Dr. Rohit Kulkarni, for coming all the way over here. Over to you. The stage is yours.
Great. Thanks very much, Neil, for the introduction, and it's a pleasure to be here. This is my first trip. I've been in Bangalore for many years, but never had a chance to visit CMC Vellore, so it's a pleasure to come here. It's fantastic to come to this campus, the Bagayam campus, and I look forward to looking at some of the other campuses as well in Vellore, and also recognize Aaron, who was in my lab several years ago. So it's great to see you too. So thank you all for coming. I know it's a holiday season, and I'm grateful that many of you took some time off to attend this talk. So, like Neil mentioned, I've got an interest in islet biology for many years, ever since I did my medical training and did some of my research at St. John's. Went on to do work at the Joslin before, subsequent to my trip to London. So for the last several years, we've been focusing on many aspects of biology, but one of them which has emerged over some of the areas that we've been working on includes RNA modifications, and I thought I'll give you a little bit of insight into what we've been doing in this emerging area. And I say emerging area because it's been surprisingly of little interest to many of the researchers in metabolism, although it's a big area of research in cancer biology, but not so much in metabolism. So we're still developing and identifying new things coming out in this area, which I'd like to share with you, to also encourage you to look at this aspect of biology which has been neglected. So with that, I just wanted to start with just press enter. So I just want to start with this central dogma, which many of you have heard from biochemistry and molecular biology, that you have DNA, RNA, and protein in sequential order. That's called the central dogma. And this cartoon was taken from Francis Crick in 1956, when he made this observation in his speech to the Experimental Biology Symposium. And he noted that in addition to the central dogma here, in this sequence of events, RNA can also talk to DNA, and RNA can talk to itself. And that was something that was proposed at the time, and it is not recognized for a long period of time. But he also made the statement that this never happens the other way, which is true. But these two aspects here, that RNA can talk to DNA and RNA can talk to itself, in the overall regulation of protein is something that has been completely neglected. And so, and you can see that there is data coming out in 1970s where they could show in hepatoma cells and viral cells there are aspects of RNA which can be modified. So if you're looking at a change in RNA, you might not see a change in the level of RNA, but because there's a chemical modification. So you really have to go into that in greater detail. So I'll go into that a little bit more in the context of this pathways of M6A. And I'm choosing M6A as one of the modifications. There are over 800 modifications of RNA, maybe about 200 in eukaryotes. And many of these RNA modifications are very well conserved. And so that gives you pause for thought that they are pretty important. So just to give you a little bit of flavor as to what exactly I'm talking about, transcripts in all cells in the body are co-transcriptionally regulated by a set of writers, which are the metal proteins. And once these methyltransferase-like proteins deposit the M6A onto the transcript on the adenosine moiety, it traverses through the nucleus on its way to the cytoplasm. And on the way, it meets the reader, the erasers, which are the ALK-PH5 and FTO. They're able to take away the transcript's methylation by acting as a demethylase. So you deposit the M6A gets erased or not erased, and then it starts to be read by readers, the reader proteins, which includes all these DF family and the IGF-2BP family of proteins, which determine whether the transcript gets exported out or gets spliced. So depending on the M6A, the transcript can get spliced, or it can get exported. And when it gets into the cytosol, it can get degraded or begins to move towards translation to form the protein. So you can see this whole thing is orchestrated very carefully by the set of enzymes called writers, readers, and erasers. And this occurs in all cells in the body, and not specifically in the beta cells, which I'm going to talk about as an example. So this is the overall sort of schematic of what I wanted to focus today. I'll go out a little bit here and there into other aspects, but give you an example of what we have found over the years. And as you can imagine, the writer complex is complex, and you can see that there are three major components: metal 3, metal 14, and WTAP, which are considered the three important components. Metal 3 is a catalytic subunit. Metal 14 is important for acting as a scaffold, and WTAP acts as an adapter subunit. And you can see all of these are integrated with multiple other proteins which are still being characterized. But the chief components are these three, which are able to deposit the M6A onto the adenosine moiety of the bases in the transcripts. So what exactly am I talking about? As I mentioned, you're talking about addition of the methyl group on the adenosine moiety of the transcript. And as I mentioned, this is very well conserved, discovered many decades ago, and this gives you the amount that is there in the RNA and why we think it's important. A couple of other points also to mention, that it's very well conserved. That makes a big advantage, because mouse and man, you can do studies in mice and they're applicable to humans, right? Because it's always difficult to do studies in human tissues, but since it's conserved, then you can do these experiments in mice. There's a very characteristic motif called the DRAC motif, which is shown here, or GGACU, and you see that this particular motif determines that it is M6A linked. And the methylation occurs in aspects of the transcript, which is the transcription start site and the stop codon, as shown in the blue here. So these are the areas where the methylation is occurring in the transcript. And we're still not reached a stage where we can do base resolution. So you really want to know exactly where the methylation occurs. But the methodology is still not reached. We can just we can just determine vast areas where there's a bump in the methylation and say that, okay, these areas are methylated. We have not gone so far as in DNA methylation, where you can have exactly where DNA methylation occurs in CPG islands and so on. So we're still lagging behind in that context, but it tells you that clearly some more work to be done. So this is just the history of the timeline here. This is when the 50s, where you have the M6A and bacterial eukaryotic total RNA, and you go all the way down to 2013, which is when my lab started to work on this. 2013 and 14, when ALPH5 was discovered as a demethylase, that's when the mass spec approaches got to be much more fine-tuned. And as you can imagine, we started working on islets. Working on islets is always a challenge, and especially because islet is a mix of cells, right? It's not a single cell, and so whatever you find in islets, you have to go and back and do it in a single beta cell. But nevertheless, Dario was a very, very talented and ambitious student in the lab who started this work. And I took a big chance because I didn't have any funding at the time. And jumping fields is always a bit tricky. But you have to do that to survive in science. And that's a note to the youngsters here that you can't focus your work only on one area. You have to keep jumping to another area, which is much more recent. Also have to learn new technologies and approaches, which makes you more competent to address the field as emerging scientists come in. So that's an important part that I did in my own career, and I'm very happy that I did it because my major focus was growth factor signaling in islets, but then we jumped to looking at RNA modifications. So in all transparency, we have already published some of this work. I'm going to give you a little bit of the flavor of what we have already shown. We think that M6A methylation of RNA plays an important role in normal and abnormal physiology, especially related to type 2 diabetes. So I'm going to touch upon two components. One is type 2 diabetes in the adults and also developmental biology. And as I mentioned, Dario was in my lab for some time. Now he's now an assistant professor in Chicago starting this year. And with all the troubles with the NIH, he managed to get his K99 award in his R01. So I'm really happy that he started his lab in June of this year. Sevim was the other fellow in the lab at the time. And I'm sure Aaron, who was also in the lab at the same time, recognizes these people. She spearheaded the work on the developmental biology, which I'll talk to you in the second part of my research talk. So this gives you just the protocol that we typically do, so that you get a flavor as to what I'm talking about. So when you talk about M6A RNA changes, you typically get total RNA from the tissue, right? So you get the total RNA. And importantly, you have to let through a couple of steps. One is you get your RNA sample, you fragment it, and then you do the IP with an anti-M6A antibody. This is the killer step because you have to get RNA. Getting RNA itself is difficult, but getting mRNA is even more difficult, right? And then you do an IP of the mRNA. So we're all used to doing protein Western blotting, but just think about doing a blot using mRNA with an immunosuppressant antibody. So that is a huge challenge in the field. But nevertheless, we stuck to it. This took us about 2 years to collect the samples from control individuals. These are cadaveric islet samples and the type 2 diabetic islet samples. So Dario was very, very patient. He collected the samples for 2 years, and then you start your experiments. It's not trivial. So nevertheless, we were able to do this study of obtaining the M6A sequencing from these samples. And so shown here is just the protocol here of how we do this for a period of time. We published 2 papers, including this one I just mentioned, but also the way to do the sequencing approach in collaboration with Chuan and Amanji in Chicago, called the RADAR. So any of you are interested, you can look at this particular approach to set it up in your own lab if you're interested. So when we began to look at these studies, the first thing that Dario did was to define the M6A methylome. So you collect the RNA, you do the M6A immunoprecipitation, and then you subject it to M6A sequencing. And then you get the M6A methylome, right? So you get the entire signature of the M6A of all the transcripts. Then you do these differentially M6A methylated gene analysis comparing the type 2 versus the controls and look at which are the pathways that come up. And you can see that you have all these pathways which are important cell cycle control, which are coming up. And then you can look at the enriched pathways where you have alterations in the peaks which are decreased, right? So you can do differential analysis and also look at those peaks which are upregulated or downregulated. And you can see that the genes which are decreased, showing M6A peaks, are all related to a number of signaling components, but also right at the top is MORI, right? So MORI comes up very, very frequently. And so we have an effort in trying to understand why methylation is so critical in the components of those transcripts which are related to MORI, and which I'll touch upon a little bit. But importantly, all these signaling pathways, growth factor signaling pathways, which we are interested in our lab. And also, you can see that if you look at the a plot of these peaks expressing a type 2 diabetes over the controls, you can see those which are hypomethylated are much more than those which are hypermethylated. So in type 2 diabetes, we believe that M6A methylation is hypomethylated. So how does hypomethylation lead to changes in the transcript behavior? I can show you in some slides. So the next thing that you can do, we can begin to look at these pathways, is to understand how it's important for beta cell biology. So you can look at these functional enrichment analysis of the differentially methylated genes, comparing the type 2 versus the controls, and you can see all these different pathways which are emerging. You can see that all of these pathways are critical for beta cell biology. You can do an additional analysis here. I showed you that I mentioned to you that these coverage plots of the entire transcript, you can plot for each one of these, control versus type 2 shown in blue, and you can see that the blue is always lower than the red shadows, and these suggest these are all hypomethylated. But there are also some components which are hypermethylated. We see the blue is dominant. So for AKT3 and FOXO1, there is hypermethylation. So I just wanted to caution here that not every methylation is hypo or every methylation is hyper. It could be a mix of each of these depending on the context of the transcript and depending on the function of the pathway. So it can go either way. And also the hypo and hypermethylation will determine how the readers will read the transcripts and send them towards splicing or send them towards degradation or sending towards translation to make the protein. So each one of these components is very well iterated and very well regulated by the reader protein. So we need to begin to make sense about what exactly this means in diabetes. But just to cut the story short, because some of this has been published, when we do the M6A sequencing, the input is the RNA sequencing. So you use the RNA as the input and then go further to look at the M6A sequencing. So RNA sequencing is what we typically use, right? Typically, everyone uses skeletal muscle, liver, adipose. Let's just do RNA sequencing and see what's going on. And this is what you get when you look at the islets. The blue is segregated from the red, but it's still not so clear. And there's a lot of variability. So these are the 8 samples from the controls and 9 samples from the type 2 patients. You can see there's a lot of variability. But look at what happens when you do the M6A sequencing. See how well clustered they are. The blue clusters are very tightly packed here compared to the red. So this tells you that just looking at RNA sequencing does not give you the right information. You need to be looking at one further step of methylation of these RNA transcripts. That allows you to segregate the disease status from the controls much better. So we really need to look at RNA sequencing with a touch of salt, but go further on to look at what M6A sequencing is all about. So we went on to do a series of experiments to address this in greater detail. And we went back into our interest in growth factor signaling. And you look at all these all these proteins with red shades are the ones which are hypomethylated in this pathway. And you can see that most of these transcripts are related to regulation by AKT and PDX1, And we know that PDX1 is the master transcription regulator of the beta cell, important for cell cycle progression, development, and insulin secretion. So you can see that doing this methylation approach of understanding the transcript regulation has given us a new insight into aspects of beta cell biology, which was not clear before. We can clearly now say that regulation at the transcript level is critical for maintaining beta cell homeostasis. And in type 2 diabetes, this goes awry and leads to many changes in what could be happening in dysregulation. But what I've shown you so far is everything is in islets, right? And islets are made up of 5 different cell types, including additional cell types like endothelial cells when you look at islets. So we have to do all the experiments focused only on the beta cells, and you can't do these from single human beta cells because it's a challenge to get enough beta cell numbers. So we have to do this in the endo-C beta cell line. So what Dario did was then knock out the writers. So if you knock out the writers, you lead to hypomethylation or reduced M6A, which is what we found in the human type 2 islets. So you begin by doing knockout of either one of them or both together. And you can see that if you do a metal 3 knockdown or metal 14 knockdown, the changes in transcripts are very, very linear, suggesting that when you do either of the knockouts, they're affecting the same type of genes. And that's why it's linear, right? Otherwise, it would be skewed. So that gave us confidence that doing the knockdowns or the knockouts in the endo-C beta H1 cells gives us a handle on what could be happening in terms of the transcript regulation, which could be similar to what we saw in human islets. So then what Dario did was he did RNA sequencing in these cell lines with the knockdown of metal 3 and metal 14, and then he intersected that with M6A sequencing from the type 2 patient data that I just showed you. So on the one hand, you have the total islets. On the other hand, you have the beta cell lines. You do the intersect, and you'd get those changes in the beta cell lines which are impacted in the islets. So the intersect will give you specifically those transcripts which are altered in methylation, but in beta cell specificity because of the endo-C beta H1. And then you begin to look at the enriched pathways, and again, you see Modi comes up again, a number of these signaling proteins and pathways, including differentiation of the endoderm, which pushes towards pancreas development. But the point that I'd like to emphasize here, that you can do all these experiments in cell lines and islets, and you intersect these data sets, and you can get a beta cell specific signature. So then one has to do the validation experiment. So you go back to the cell lines, and then you do a knockdown of either metal 3 or metal 14, and then you do simple experiments where you stimulate them with IGF-1 or insulin. In this case, I'm showing you IGF-1, and you can see in the scramble you have phosphorylation of the insulin receptor, phosphorylation of AKT. But we'll see what happens to the cell line which has a knockdown of metal 3. Phosphorylation of the insulin receptor, IGF-1 receptor is blunted, and AKT is blunted as well, as it's quantified at the bottom. So if you do a knockdown of metal 3 and metal 14 in the cell line and stimulate them with the ligand, with insulin or IGF-1, you have blunted phosphorylation, which is telling you exactly what you find in the islet. So it is validating what you see with a knockdown of metal 3 or metal 14. Then you can do additional experiments to begin to look at cell cycle control. So shown here is a cell cycle experiment where you have scramble, a metal 3 knockdown, a metal 14 knockdown in the cell lines. And then you can see there is a distinct difference in the cell cycle changes. The G0, G1 goes up from 69 to 72 to 75. And you can see that the S phase is reduced, G0, G1 phase is increased. So this suggests there's a cell cycle arrest. And this can be quantified at the bottom. So it tells you that interfering just with the methylation, you're already influencing the cell cycle control of the beta cell. You can also do other experiments. Obviously, you would be interested in looking at glucose-stimulated insulin secretion. So you can see that compared to scramble, where you have insulin secretion is increased by 16.7 millimolar compared to 3.3. And this is blunted when you do the metal 3, metal 14. This blunting is mostly because of increase in baseline glucose secretion. glucose-stimulated insulin secretion. Insulin content goes down as well. So you can begin to show the effects of these changes in methylation known as cell cycle control, as well as insulin secretory defects in the beta cells. So what about in vivo? When we submitted this paper, we showed all the data on human islets and then the cell lines, and one of the reviewers said, you should do a mouse model. So it is very puzzling to us. You showed all these data on the humans, and they're asking us to go back to the mouse. So in any case, we did a knockout of metal 14 in a mouse model. I won't go into great detail because it's all published. So these mice developed hyperglycemia by 4 weeks, reduced beta cell mass, reduced beta cell proliferation with very few insulin granules on electron microscopy. And they showed all hallmarks of over diabetes by 8 weeks. So we could show, with all the data that we show in the human islets and also the cell lines, you create a mouse model in vivo by knocking out metal 14, which reduces the M6A levels, and you have the entire phenotype of type 2 diabetes, suggesting that this is very critical. So we went back to the data here, and this was published more recently last year by SEVM, begin to understand some components of developmental biology, right? I showed you when you change these methylation patterns, the MODI keeps coming up. We have a separate study in that context going on. The other component that emerged was developmental aspects of pancreas, and so initially, SEVM started doing these experiments by first establishing alterations in mRNA stability of these beta cell genes. So, to give you one example of the validation experiments, you can do studies where you look at the decay of the stability of the transcripts, right? If you remember the pathway is showing you, M6A is important for splicing. It's also important for degradation of the transcript. It's also important for translation components and decay of the transcript. So you can do experiments to establish that. So given here are 2 examples, where you do a knockdown of metal 14, shown in green, and we see that there is the degradation of glucokinase and IGF-1 receptor are affected. So these appear to decay faster. So shown here is time after actomycin D treatment, which tells you the decay of that particular transcript and proteins. The IGF-1 receptor decays faster compared to control, and so does glucokinase. So interfering with the M6A transcripts, you can see that there is alterations in the half-life of these transcripts, leading to reduced accumulation of the proteins. So many people argue that when you're looking at M6A or the transcripts, you're not really looking at the protein levels. But here's an example where we really look at protein levels and show there is a distinct change in protein. So that's the summary here. I just want to emphasize that when you look at the M6A pathway, it appears to affect multiple transcripts. So in our approach of reductionist biology, some of us are focused only on some aspects of transcripts, right? Only some transcripts. So for example, saying only glucokinase is affected, or perhaps only glucose transporters are affected, or perhaps one transcription factor like MAFA is affected. But the M6A approach argues that multiple transcripts are affected at the same time in type 2 diabetes. That would make sense because teleologically, the regulation of the transcripts all occurs together. So why would a cell have a distinct effect only in one protein, but not in multiple other transcripts, which are being regulated at the same level? So the argument of the M6A approach is that you're identifying multiple transcripts, which is affected by pathophysiology, which leads to the defect in the beta cell. So I'll come back to that a bit later. So the second aspect I just want to touch upon is about pancreatic cell differentiation. So when we did these early experiments, as I mentioned, some of the things that kept cropping up was MODI and pancreas developmental components. So all these transcripts which are important in pancreas development kept coming up in the enrichment pathway analysis. So we decided to do this in a more systematic manner. And And Sevim really spearheaded this, which took us several years to complete for obvious reasons, because getting pancreas from humans at different developmental stages is not very trivial. Doing it in mice is great, but then you have to do this translational component of showing it in humans, right? So that is the challenge. So shown here is, some of the transcripts which are altered in the context of type 2 versus control, in the context of M6A methylation. And you can see many of these transcripts kept coming up. So we went on to do this experiment initially by formulating this hypothesis that M6A is somehow important in regulating it's a very broad hypothesis, which we have then since narrowed down. And all this is published last year. So I won't go into great detail here, but those of you who are working in this area know how long it takes for us to consider human pancreas development. 9 months, right? It takes 9 months. But we now know from all the stem cell approaches that you can reduce to 45 days. So we can now take a blood sample from an adult or a skin biopsy from an adult, and then you derive adult adult stem cells, and then you differentiate them towards pancreatic progenitors and all the way to beta-like cells in about 45 days. But we're still not there yet, in the sense that you've got most authentic beta cells are still not available. There's still some issues which we have with the cells. But we have to also jump down to the mouse model for some in vivo experiments, because you can't do these experiments in humans for obvious reasons. So we've used PDX1 Cree-driven animals, where you do a knockout of metal 14, NGN3 driven Cree-driven animals, and INS1 Cree driven animals. So these are the 3 different Cree-driven developmental animals that you can use. PDX1 Cree relates to this age in mouse development versus human pancreas development. So shown here is PDX1 Cree and NGN Cree and INS1 Cree all the way down here. So you're able to develop these models to see what it relates to human developmental patterns, but also more importantly, it gives you a handle on when exactly is methylation important, at what stage of the development is methylation important, right? So then we had to do the hard work of collecting these pancreas, and obviously these samples, we are fortunate to get these samples, and it's unfortunate for the patients, obviously, but we make use of these samples from a large collection of pathology departments across the US, but also from NPOD, which is the network of pancreatic organ donors, which allows us to use some of these samples for clinical research. So shown is a list of the samples that we use in prenatal and postnatal. And we began to look at the expression patterns here on the metal 14, metal 3, and ALPH5, which are the writers and readers. Shown here is an example of what Sevim did, looking at the expression of metal 14, co-immunostaining with insulin and DAPI at 12 weeks, all the way to 41 weeks. And you can see that there's a large mix of the signatures, and you have to quantify this over time. And that is shown here. And Sevim could clearly show that metal 14 expression increases from prenatal age of 12 all the way to 48 weeks, suggesting that the M6A methylation pattern occurs linearly, increases over a period of time. So methylation increases as the pancreas develops from 12 to 48 weeks. That would suggest that this methylation is critical during pancreas development. And then you can begin to look at the postnatal levels. And there she found it was very much flat. So postnatally, when it peaks at that level, methylation of the cells begins to show a plateau, suggesting that, again, M6A is critical during early development, but not after development. So that pushed us to begin to understand this in greater detail as to how exactly metal 14 is playing a role during early development of the pancreas, and begin to understand in sequential order as to how we could look at this more carefully. We took advantage of the other data sets which are also available in RNA sequencing, which have been published by other groups. And we could go back to our studies and begin to see that there is a difference in the methylation patterns between beta cells, which produce insulin, and glucagon-producing cells. So if you look at the fetal beta cells versus adult beta cells in terms of gene expression patterns of all these different transcripts, the fetal versus adult beta cells shows a certain pattern which is different from the alpha cells. So there's a distinct difference between beta cells and alpha cells in the context of gene expression patterns of these M6A regulators. So, Seven went back to looking at expression patterns of these M6A regulators in human fetal versus adult, and as I mentioned, first focusing on the beta cells. So shown here are gene expression of the writers, the three major writers, and you can see there is some amount of difference here comparing the fetal versus the adult. The most distinct component is ALPH5, which is a demethylase, and some of the readers. But very interestingly, we didn't see this kind of regulation except for maybe metal 3 in the alpha cell. The alpha cell regulation appears to be quite different. So this again argues that the beta cell is much more dominant in the context of M6A regulation versus the alpha cell. And even in our type 2 diabetes studies, we focus on the beta cell with very little difference in the context of the alpha cell. But we also have a very separate effort on the alpha cell in the context of dynamic changes in regulation when you're subjecting them to stimuli like arginine or just glucose itself. You know that glucose also regulates glucagon secretion. So when you look at the secretagogues, the alpha cell behaves differently, but not during developmental patterns. So this is just a cartoon, as I mentioned to you before, looking at how we've reduced the time frame of 9 months of these human pancreas development to about 5 weeks, where we can look at regulation of different aspects of beta cell biology. And some people argue that what we're looking in this truncated manner is not really reflects what could be happening in vivo, but this is the best that we can do right now. So we set this up this many years ago. I've been involved with stem cell biology now, right from 2004, 2005, actually, when the initial effort was started by, once we got to know the Yamanaka factors at the Joslin. So we have developed this approach at the Joslin pretty significantly now, and we can develop immature beta-like cells within 40 or 45 days. And so Seven began to look at the pattern of metal 14, looking at this description of these different progenitor cells all the way to the beta-like cells, and she could see that metal 14, as was evident from her immunostaining of the pancreas sections, even during the in vitro differentiation, showed this pattern of increasing over a period of the differentiation stages. So showing this again from stage 1 to stage 6, there's an increase in metal 14. So I showed you the immunostaining pattern also. The prenatal showed an increase in metal 14, and this is replicating in the in vitro differentiation approach, showing an increase in metal 14. So this gives us the confidence that we can begin to use these progenitor cells and differentiated cells for dynamic experiments to understand how metal 14 M6A is important, which aspects of developmental patterns. So she also referred to, as I mentioned, can take advantage of the publications from previous data sets which have been published. We can begin to look at those transcripts which are regulated by methylation from our data set that was published in the type 2 diabetes paper, and look at those components by revisiting these data sets which have already been published. And what she could show was that the stem cell-derived beta-like cells or stem cell-derived alpha-like cells, when you look at the stage 5 to stage 6 the red is showing increased expression. So metal 14 is very distinct, that it shows an increased expression from stage 5 to stage 6. But the alpha cells don't show that much of a pattern. ALK-BH5 is the other one which stands out, showing that from stage 5 or earlier stages all the way to stage 6, very distinct increase in expression. So this tells us that metal 14 and ALK-BH5 are two components which are key in M6A regulation during early differentiation of the beta cell. And it's quite different from the differentiation of the alpha cell. This again provides data from orthogonal data sets published already in the field, which is a major advantage these days to address these, because you can't do everything yourself. So how exactly does M6A levels change during in vitro differentiation? So she looked at some different genes which we are interested in. I mentioned to you about monogenic diabetes. PDX1 we know is one of the genes which is important. And here you can see that in PDX1, there seems to be a jump from stage 0, stage 1 to stage 3, and then it plateaus off. So there seems to be a sudden increase in the expression of the M6A levels in the transcripts for PDX1, which is very, very interesting. So something seems to happen between stage 2 to stage 3, which triggers this very sudden increase in M6A. But there are others like NKX6.1, where you have the pattern which is much more gradual. So you can see that S4 is much lower and it reaches maximum by S6. So you can see the difference between PDX1 and NKX6.1. And insulin itself, again, has a distinct pattern where you can see it comes up only at very late stages. So the M6A regulation of these transcripts becomes important at the different stages of differentiation. And one can begin to look at this in greater detail, how exactly the methylation pattern is being regulated and at different stages of the transcripts and the expression of the protein levels. So this gives you an added level of insight into the regulation of these transcripts, which was not known earlier. This is just again the cartoon that we show, doing the profiling by M6A immunoprecipitation, and you look at the M6A abundance in these the exons, stop codon, the 5' and 3' ends. So then, what can we do with this data? So we can begin to do dynamic experiments of interfering with the metal 14 right in the embryonic stem cells, right? You take the embryonic stem cells and then begin to ask what happens if you knock down metal 14 or metal 3 or ALK-BH5 and then let it differentiate towards the pancreas progenitors and then see how it develops to the different transcripts. So this gives us an additional approach to do a dynamic test using this shRNA vector approach. So shown here is just the details of the method she used. She used the inducible human embryonic stem cells, these two different cell lines which are available now commercially, MEL1 and H1. And then you can begin to do these metal 14 knockdown approaches using the doxycycline inducible and generate these knockdown cell lines, 3 different cell lines of the MEL1 and 3 different cell lines of the H1. And then begin to look at what exactly happens in the context of differentiation of the cells. So what is interesting to us was that when you interfere with metal 14, they did form the embryoid bodies. So there was no problem with developing the embryoid bodies when you interfere with M6A, which then tells you that formation of the endoderm, the ectoderm, and the mesoderm is normal. It doesn't really interfere dramatically with changes in early differentiation. But there are things which begin to emerge. So you can see that clearly this is metal 14 is what we have knocked down. When you do a metal 14 knockdown, very curiously, WTAP also goes down and ALK-BH5 goes up. So this is telling us that when you interfere with metal 14 alone, the other regulators are trying to compensate in some ways towards maintaining the M6A pattern. So that would be one interpretation of the changes in ALPH5. But we're still trying to understand why WTAP is reduced when you interfere with metal 14. So there seems to be some kind of a crosstalk between WTAP and metal 14 and metal 14 and ALPH5, which is not very surprising in an equation where you see that you interfere with one end, the other end perhaps regulates to maintain the equilibrium. And then she's gone on again. I'm jumping a lot of steps here for lack of time, but doing all these knockdown experiments and looking at those genes which are downregulated versus those genes which are upregulated in the context of, especially in the context of ectoderm and the mesoderm gene expressions. And then you can begin to look at which are the enriched pathways which are being affected. Again, we go back to the cell cycle control, G1S transition, mitosis, which are key components in cell cycle progression. And you can also see that those genes which are upregulated, so on the left are the ones which are downregulated, and the ones which are on the right are the ones which are upregulated. And very interestingly, we see a number of pathways which are important in differentiation, but one of the pathways which we're interested in is the neural crest differentiation, which we're pursuing this in some ways. But this again gives you an insight into why M6A methylation is important in early pancreatic differentiation and how that could be related to understanding pathophysiology. So in the last few minutes, I just want to give you a couple of examples. I know this might be a repetitive for the next set of slides, but with all the data that we have generated now in the when you knock down the metal 14. So I showed you data starting with the human pancreas samples, the prenatal and postnatal, showing you the pattern of the M6A regulation. We went back to an in vitro differentiation model. We took the ES cells and then interfered with the M6A metal 14 knockdown, and we showed how the methylation patterns mimics what we showed with the immunostaining approach. And then we did a dynamic testing of these, taking the ES cells, then doing an early knockdown of the writer, interfering with M6A methylation, and then begin to see what are the transcripts which are involved. And then we can begin to pick all these transcripts individually, and then begin to see how exactly is being regulated. So this is what Sevim did here. So she went back to the initial the equation that I mentioned. You have the mRNA, then you have the writers and the erasers, where deposit the M6A, and then the readers will read where exactly the M6A is being deposited, and then it begins to, that's like a traffic policeman, it begins to move it towards translation or stability or decay or splicing or nuclear export. And we can begin to each transcript how each one of these begins to move towards one or more of these components in the end. So we started with stability. So she took one example where you've got these endo-C VERH1 cells, which have been knocked down with metal 14, and see which of these transcripts. So she looked at INSM1 and SOX4, which are important in early differentiation. And she did an actinomycin experiment to see how exactly it regulates when you do the knockdown. And she could show that this is the control where you don't see a difference in the scramble versus the knockdown. You can see that for this particular transcript, when you do the knockdown, there is an alteration. There's a faster decay of this transcript, right? When you compare it to SOX4, again, there's a faster decay. So you can see that these transcripts decay faster, and so the protein formation is affected because of the faster decay. And you can do this for multiple transcripts. So stability is one example. And then you can go back and look at what could be happening to export, right? You can again go back to your cell lines and look at specific transcripts and see how they're exported out by doing experiments with cytosolic versus nuclear ratio. And this is again a control which is not different between the knockdown and the controls. This specific when you do the ratio with the cytosolic to nucleus, you can see it's increased, suggesting that it's much higher in the cytosol compared to the nucleus. MIS2 is also showing the same pattern. So does the GIP receptor. So you can see that all of these are being exported differentially when you do a knockdown, suggesting that M6A is important for the nuclear export of these transcripts. And then you can go back and see what happens to translation, right? Translation is another example. So again, you do the knockdown with the control, and you do a 35S methionine incorporation to look at the translation. And you can see that compared to 0.5 or 1 hour of the incorporation experiment, there's a significant decrease in these transcripts, which suggests that there is an alteration in the translation. You also can do other experiments. I'm just giving you some other suggestions here that you could do. You can look at polysome-associated mRNAs. So RNA biology is emerging as a very big player now in metabolism. And many of these assays that I'm showing you here gives us handles on doing additional experiments to understand how M6A is important in the context of export translation decay. But shown as one more example is polysome profiling, where you can see that when you do a knockdown to metal 14, there's an alteration in the polysome signature. You can see that the number of polysomes are much lower when you do the metal 14 compared to the control. You can then begin to pick of all these polysomes and begin to see which of these transcripts are being affected, right? So that's how you do polysome profile. So in the last 1 or 2 slides, I've showed you a little bit of data today on some aspects of RNA biology, where we think that one of the modifications, which is adding the methyl moiety adenosine, is important in regulating RNA, the way it gets translated or gets degraded or gets exported or gets spliced. With those data, we came up with 2 sets of observations. One is that the transcripts can affect multiple pathways, right? So shown here are a number of pathways which have been shown. If you look at the literature over the last 40 years, each one comes up with one particular area of interest. So you have glucose sensing, you have mitochondrial dysfunction, you have exocytosis, ER stress, dedifferentiation. So we believe that each one of these is not a separate entity, but rather all of them can be clustered by M6A methylation, because these transcripts gets turned over a period of time and affects multiple transcripts, each of them with different pathways. So we don't believe that these are different pathways in patients with diabetes, but rather all these pathways are affected together by transcript regulation. And this is the concept of the transcriptome turnover as opposed to protein turnover. We're used to protein turnover, right? So the concept of transcriptome turnover is that these transcripts are being altered sequentially or in multiple ways in waves, right? And I think this makes sense for the cell to be more energy efficient. Why would a cell make a protein all the way to the end and then realize it made a mistake and start all over again? But perhaps it gets regulated at the transcript level. So as soon as it realizes it's made a mistake, it stops there. It doesn't go all the way to the protein and then come back, right? So it makes sense for us to learn more about the transcript regulation to allow for us to understand homeostasis of the proteins in the long term. So somehow this has been neglected in the field, and we think that looking at this transcript turnover much more carefully will be useful. So for all the trainees, when your PI tells you, let's forget the RNA, you have to go back and say mRNA modification is important because it plays a big role in translation, and decay, and degradation, and splicing, all of which can affect protein function. So we are doing a number of different experiments. Obviously, I'm always very excited with these new approaches, and it gives us a handle on trying to understand in greater detail about how M6A may be important. In addition to M6A, we're also interested in pseudourylation. Pseudourylation, if you guys remember, was something that was used to develop the COVID-19 vaccine, right? So it was it was a pseudo-urelation of that transcript, which allowed them to develop the COVID-19. So it's got its own therapeutic applications as well, which from a different angle. So that would be an important thing to keep in mind as well. So I just want to stop here and thank the people. Again, I just showed you data from what Sevim and Dario did, and all of the lab members and many of our collaborators. And I'm sure you'll recognize this guy there, this is just one of the images we took from a long time ago. This was during the COVID times, and Aaron was there in the lab, working hard, coming in even when there was a lot of restriction. But the exciting times, and we hope you'll come back again. This time hopefully it will not be restricted work. But I'll stop there and thank you for your attention. I'll be happy to take questions.
Thank you, Mr. Rohit, for that absolutely mind-blowing, wonderful talk. I think we have a range of people over here in the audience, those who are some who are molecular arena, genetics, others who are clinicians, and some who are probably learning how to spell the word mRNA. So that's a vast diversity over here. But anyway, over to the audience for your questions. And I think this is a huge opportunity for you to ask questions, interact. Of course, we're all inspired by such a mind-blowing talk. Over to you.
Very nice talk, Prof Kulkarni. And I have two questions. One is that this metal 14, whether you found in diabetic patient that it is either not there or it is low. Therefore, the whole M6A is changed. That is one question. The second one is why you did not make a knockout instead of doing a knockdown, because it's not developmentally critical, as you showed in the mouse knockout experiment, that they are not embryonic lethal. So why you have not done in the cell line the knockout experiment?
Yeah, no, good questions. Yeah, so I skipped some data initially. So we did due diligence. We first took samples and looked at the expression of metal 3, metal 14, LBH5, FTO in samples, and we could show that metal 14, metal 3 are downregulated in the islets, right? So that would suggest that M6A is also going down. So we report that in the paper, so it's already there. We also did experiments. The other question is insulin regulating M6A? Is glucose regulating M6A? Are amino acids, fatty acids regulating M6A? The answer is yes to all. So glucose does regulate M6A, but again, it's context dependent. Very high glucose reduces M6A levels. Insulin is also important in regulating M6A levels. So many of these components argue for a role in metabolism, right? Fatty acids, glucose, insulin. So we did do a decrease. So we did a knockout in the mouse model to completely suppress M6A. But in the cell lines, we decided to do a knockdown because we didn't see a knockout in the human tissues in type 2. It was reduced, not completely gone away. So that was the rationale for doing a knockdown rather than a knockout.
And do you see in those patients who are having this low level of metal 14, like should we also screen people who come with type 2 diabetes for looking at the level of this, that whether this is dysregulated or not?
Well, from our studies, we would argue that looking at the M6A regulators will help, because it allows you to then begin to look more specifically at those transcripts which might be involved, which may give you a better handle on therapeutics further down the line, right? So if there's no change in RNA, if there's no change in mRNA, there could still be changes in M6A levels. So that's the major point I'm trying to make here. If you just take a tissue, you take an adipose tissue or islet, you look at the mRNA for some protein. If there's no change, I would recommend to go one step further to do an M6A on that, because there's no change in mRNA, but the modification is changing, and the modification can still have an effect on the protein. And that's why we call this epitranscriptomics, because there's no change in the overall mRNA, but there's a change in the modification. So just detecting mRNA will not help. You have to do sequencing to look at the M6A.
And can we supplement to bring its expression to the normal so that we can have a normal M6A profile?
So we have done some experiments. Again, we've published a couple more papers, one in cell metabolism on adipocyte biology, and one in white adipose tissue as well, where you can do the rescue. So you can go back and put in metal 3 and expand and show an increase. In fact, in type 1 diabetes model, which we published in Nature Cell Biology last year, Dario did a very clever experiment where he took the NOD mouse model and he could overexpress metal 3 only in the beta cell, and he could show that he could rescue the diabetes. So it is possible. Therapeutically, it's possible to show an increase in metal 3. There are also some small molecules which you can use, which can increase M6A levels. So you can see that therapeutically it's developing, but still a lot more needs to be done.
Thank you.
Other questions online? Maybe not.
I have a question.
Yeah,
In your initial part of your presentation, you used the term maturity onset diabetes of the young as a generic term. So when you talked about the impact of writing MP6, wasn't it? I mean, because there are several types of voting, but you use it like a generic term. So I was wondering why you did that, or is that was the impact kind of on multiple various kinds of transcription factors that it might be seen right across the board for many types of voting?
Yeah, no, I think the MODI comes up as a generic term in the pathway enrichment analysis terminology. But we have gone one step further. But we took the hint about that's the way it comes up on the geo terminology. But we did show, PDX1, for example, HNFs. We also have an interest in MODI 8, CEL. But we are interested in PDX1 glucokinase and one of the HNFs, distinctly looking at what happens to the M6A approach. But that is a generic term used in the GO terms.
And of course, I think the other very important thing is we always presume that MODI is a very beta cell defective disorder. But I think from what you have shown, it perhaps also has impact on the resistance level as well. You said through IGF growth factor, other ones like that. IgF binding protein.
Yeah.
Well. So it's not truly a pure beta cell T3 disorder, as in many of them aren't.
Yeah.
Is that,
yeah, no,
that's a message that's coming across quite
Yeah.
strongly.
Yeah, absolutely. You're right. I think we have simplified it like it's completely just beta cell specific, and it looks like it's much more than that.
Yes.
Hi, sir. So I have two questions. So one is, so you have observed that metal 3 and metal 14 is getting down-regulated in beta cells of diabetic patients. And you are saying that when you're upregulating those, you're seeing a reversal of the phenotype. So have you checked at the genotypic level in those patients, what is causing the down-regulation of, or is it some environmental factor or habits, down-regulation of metal 3 or 14?
Yeah, that's a tough question to answer, I think, because we still don't know. Obviously, I think what causes that is the challenge. I guess one way to address it would be to look at GWAS, right? So in the type 1 diabetes model, which we have published, we focused on metal 3, and we observed that metal 3 changes transcripts which are related to the oligoadenylate synthetase genes, the OAS genes. And OAS genes have been identified as GWAS genes for type 1 diabetes. And so the question there is, which is it? Is it really a defect in the gene, or whether it is metal 3 which is causing that? The GWAS tells us that, okay, there's a SNP in the OAS gene, right? That really associates with type 1, and it's caused by the SNP in OAS. But we've done a lot of studies on metal 3 to argue that changes in metal 3 is the precursor to changes in OAS gene behavior, which leads to beta cell demise in type 1 diabetes. So even if you have OAS as a GWAS gene, one can argue that metal 3 is more important, but doesn't answer your question as to what is causing the metal 3 change in the first place. We don't know that.
Is there any mutation or common polymorphism in the promoter of the gene? In those patients, have you checked?
We have checked. There are some which are close to enhancers and promoters, but nothing which stands out distinctly. That's why we feel that maybe these regulators were highly conserved, but maybe they're also somewhat redundant, because the writer complex is got like 15 proteins. I focused only on 3 of them. So it's possible that even if you lose one of those, maybe there's redundancy, and they might be okay to be lost in some ways. But their function is important in the context of regulating key transcripts in certain different pathways. The other thing I want to mention also, we published in Nature Communications recently, is if you do a knockout, so if you interfere with M6A in 2 tissues, let's say brown adipose tissue and white adipose tissue, and you have that same transcript in both the tissues, but M6A regulates brown adipose differently compared to white fat differently. The same transcript, but 2 different tissues. One is hypomethylated, the other one is hypermethylated. So you see, it's context dependent and tissue dependent. The same. So you take the same transcript from the beta cell to the alpha cell, the regulation will be different. So we're still trying to understand what that means, but I think it would argue that it's regulated to a very high level, making the tissue behavior more important than just a generic sort of effect on the multiple transcripts, right?
Thank you, sir.
Any questions from those of you online? Would you like to ask some question? Okay. So with that, I'm sure there will be other people who will be a little more refractory towards standing up and raising their voice in the audience, but you're most welcome for a cup of tea to further your knowledge and interact with this amazing personality who's given us this talk today. Let us stand and give him a hand. Thank you, Rohit, and we certainly look forward to long-term interactions with you from CMC Vellore, from CSCR, and all of us within the audience and beyond. Thank you so much. I think I must do my introduction backwards, because Rohit is in fact done his undergraduation from the Bangalore Medical College and also his post-graduation from St. John's Medical College. So there are connects over there as well. Thank you so much once again.